सेल कल्चर लेबोरेटरी में काम करते हुए समटाइम्स हमें कुछ ऐसी सिचुएशंस के साथ जो है काम करना पड़ता है दैट वी नीड सेल्स फ्रॉम सम अदर लेबोरेटरी और वी have to send cells to some other laboratory. That is kind of exchange. और अगर commercially देखा जाए तो cells are transported on routine basis when you purchase the cell lines from any commercial source. लैब में काम करते हुए अगर हमें सेल ट्रांसपोर्टेशन के हवाले से कुछ ज़रूरत पेश आती है तो हमें कुछ पैरामीटर्स का ख्याल रखना है जिनके बदौलत सेल्स जो हैं ड्यूरिंग द ट्रांसपोर्टेशन they are not going to be dead, they are not going to be damaged. पहला जो इसमें फैक्टर है द ग्रोथ. हमें अगर सेल्स ट्रांसपोर्ट की तरफ लेके जाना है वी शुड प्रिपेयर दो सेल्स फॉर ट्रांसपोर्टेशन and we need to grow those cells up to the required culture limit or to the confluent level in that flask. 25 centimeter square flask is routinely used, and now if these are monolayer cells or these are the suspension cultures, keeping in view we need to grow these cells up to confluent level. Once the required culture condition is achieved, then we need to ensure that during the transportation period the media should be, the culture medium should be sufficiently rich in nutrients. It should not be like exhausting. Its pH should be near normal, which is required for the cell culture. For that purpose we ensure that once the culture achieved or is at the required density level, we replace the previous or older culture medium with the fresh culture medium. Then, to ensure that there is no leakage during the transportation, there is no loss of culture media, we are going to properly seal the flasks, but keep in mind flask should be sealed, but the ventilation के लिए जो कैप में कुछ फिल्टर पेपर लगे होते हैं they should not be sealed, otherwise suffocation जो है वो डेवलप हो सकती है फ्लास्क में. फ्लास्क को सील करने के बाद एक लीक प्रूफ प्लास्टिक बैग में सील किया जाता है या फिर किसी दूसरे कंटेनर में, ताकि ड्यूरिंग ट्रांसपोर्टेशन अगर ब्रेकेज या डैमेज हो गया फ्लास्क को तो जो हमारे लाइफ सेल्स हैं दे शुड नॉट बी स्प्रेड इन द environment. After that, these sealed culture flasks, they are placed in insulated container. Ab insulated container will maintain the temperature. It will not make it happen कि during high temperature अगर हम flask को transport कर रहे हैं और low temperature atmosphere में हम transport कर रहे हैं तो जो rate of evaporation है उसको minimize किया जा सके. सबसे ज़्यादा ज़रूरी चीज़ जो labels हैं, that the packet, the box or the parcel should carry a clear cut label of biohazard material, because living cells हैं, they can be biohazard level उनका हो सकता है. So we need to label them so that the people who are handling these boxes during the transportation, they should know the to take all those measures which are required for handling a biohazard box or material. Frozen cells can also be shipped. जैसा कि हमने cells को cryo preservation की तरफ लेके गए, उन्हीं cryo preserved cells को हम transport कर सकते हैं, taking out from the liquid nitrogen, and for a short time we can maintain the cooling temperature by using the dry ice. We shift those vials containing the frozen cells in the boxes, insulated boxes that contains the dry ice. The dry ice is going to maintain the temperature during the shipment period, and as soon as you receive the frozen cells in the shipment, take immediate action to shift those vials wherever and whatever is the appropriate temperature for their storage. If you are interested to establish culture, then you, keeping in view the conditions, situations, you can proceed for establishing the culture from those frozen cells जो कि हम पहले डिस्कस कर चुके हैं.